Hey guys, welcome back to our last set of notes here for Chapter 5, our introduction to metabolism. So we left off last time talking quite a bit about our lipid bilayer and how we can kind of move stuff through by diffusion. So we're going to keep talking about the lipid bilayer and a little more about transport this time. The lipid bilayer for our cells is what's called semi-permeable or selectively permeable, which means the membrane is picky about what it lets into the cell, which is nice for us. We don't want our cells just letting everything into the cell. It's the ability of the cell membrane to control which substances and how much enter or leave the cell. This is going to allow the cell to maintain a difference between its internal environment and the extracellular fluid. It's like homeostasis on the teeny tiny cellular level. This is nice because it's going to supply our cell with nutrients. We can also remove waste, and we can maintain the correct volume and pH of our cell. Water is going to diffuse across the membranes by osmosis. Oxygen and carbon dioxide pretty much dissolve straight across. But other than that, we're really picky about what we let in and how much we let into our cells. So here is this kind of cool little diagram where our lipid bilayer exists. This is the inside of our cell. This is the extracellular fluid. Gases, so let's see, probably carbon dioxide, oxygen gas, and a water molecule. They're not really charged, and they can pretty much just kind of shoot right through here. This is a bit of an oversimplification because this guy actually has to find a channel because he's charged. But don't tell anybody. I won't tell anybody. It's okay. This is a good book. They're just keeping it basic for you. Now, if you're a charged molecule or a big molecule, you can't just shoot right through here. Like, look at how big glucose is. This is not going to happen. He's not going to make it through this membrane. So we got to find some other strategies to get guys like this and guys like this, top secret, into the cell if they can't shoot straight through like oxygen and carbon dioxide can. So when we look at the concentrations of solutes that are separated by this barrier, there's a few different terms with respect to tonicity that you have to learn, and they're super easy. You've done it in Gen Bio. It's isotonic, hypotonic, and hypertonic. When you're separated by a membrane, there's going to be a gradient in solute concentration. So if the solute concentration outside of the cell is the same as the solute concentration inside of the cell, it's called isotonic. If the solute concentration outside of the cell is very low and we have more solutes inside the cell, we call this hypotonic. And then a hypertonic solution has a high solute concentration outside of the cell compared to inside of the cell. So know that on their own, none of these three really stand alone. They require a cell and an environment, and they're all in relation to the cell. That's an important point to kind of understand for those. And so the example that really everybody uses for this is red blood cells. Red blood cells in an isotonic solution, we have, your 0.9%, I think, saline for a human. We have 0.9% solutes in the fluid. We have 0.9% solutes in the red blood cell. So a little bit of water moves out, a little bit of water moves in, but they all keep this kind of donut shape and everybody's happy. That's normal, healthy, everybody's in good shape. This one's not good. This is a hypertonic solution. There's way too many salts out here. So the water moves from inside the red blood cell to the environment. And as that water moves out to try to make these solutes even, these red blood cells kind of shrink and shrivel down. They don't do their job anymore. That is bad news for whatever human has that blood. Now this guy is the opposite problem. We are hypotonic, which means there's hardly any solutes in here. This is what would happen if the IV in the hospital weren't saline, if it were just pure water. You pump a bunch of pure water in there and there's no salts in the water, then there's more solutes inside this red blood cell than there are outside, so the water moves in to try to make everything equal, and the red blood cell swells from this nice little biconcave disc donut shape into this large sphere where it stinks at its job now because its surface area is all messed up, and if it swells too much, it'll actually burst and spill its contents into the extracellular fluid, and so the human who's in this situation is equally bad as this situation here. So, homeostasis, right? Goldilocks and Three Bears, too hot, too cold, just right. We want to be just right with everything. Hormones, salt concentrations, semi-permeability, we got to be just right. That's what biology is, okay? So, here we go. Turgor. The pressure exerted by a volume of fluid against the surrounding structure. Just think water pressure. I mean, turgor is a cool word and everything. You learn turgor, but all turgor means is water pressure. When a plant cell is full of water, that water exerts a pressure on that cell wall, and then it doesn't change volumes easily. The plant is strong and tough and stands up straight. If all the water gets out of that plant and the plant's super dehydrated and dying, the plant's going to wilt, it's going to be weak, it's going to be easily injured. So turgor pressure in plants is a great thing. Osmotic pressure is the amount of turgor that can stop water from diffusing, or the amount of pressure to prevent osmosis, which is the movement of water. So that's what's going to keep those cells strong in plants, is once they've reached osmotic pressure. It's going to keep cell walls really strong because they're full of water. So it's good to have those things in cells. When we start to look at transporting things, we have a lot of types of molecules and ions that can cross a lipid bilayer, and they need some help from transport proteins. And transport proteins are very specific. You've got a calcium pump, and he just works with calcium. You have a water pump, and he just works for water. And glucose just fits glucose, and on and on and on it goes. Ions, these large polar molecules, they need some way to cross the cell membrane, and they have a couple options available to them. There's passive transport, and there's active transport, and it kind of depends on their concentration gradients which one we're going to have to use. So let's start with passive. We call passive transport passive because it requires no ATP to get the job done, no energy. It doesn't cost us anything. It's driven just by the concentration gradient that already exists from our solute. So that's one type. Now there's a specific one called facilitated diffusion, and that is a type of passive transport where we're still not using ATP, we're still powered by the concentration gradient, but these guys need a door to get into the cell. And so we use a gated passive transporter, which is really just basically a channel that changes shape when a specific molecule binds to it. So you might have a channel for water, and if glucose bumps into this channel, glucose can't get into the cell. But if water bumps into that channel, it'll shoot water straight through. It doesn't take any energy to do this. It's just a door letting water, who wants to come in anyway, to get into the cell. That's facilitated diffusion. Passive, it diffuses in. Facilitated, it just needs a little bit of help from a door to get that job done. And those doors are made of, guess what? Protein. You got it, because this is biology class. Okay, so what do we got here? Oh yes, this is facilitated diffusion. So we've got a protein. He's not that fancy. He's a channel. Glucose bumps into him and he says, today is your lucky day. I happen to be a glucose transporter. So the extracellular matrix side pinches down just a little. The cytoplasm side, I guess you could say, relaxes just a little bit, and he kind of shoots glucose through. Now he didn't go this transporting channel. He didn't go through any massive mind-boggling changes, so it didn't cost any ATP to do it. He just kind of closed one side of the door and opened the other a little bit, and glucose did the rest. And here in a minute, another glucose is going to bump in, and he can do this job again. And it's fine, because he's not going to run out of energy, because it doesn't take any energy. Not a big deal. So, so much trickier. Active transport. We want to closely regulate the concentration of certain ions and we can't just let them go through where they want to go through. So we are going to use active transport, which requires energy input. Most of the time, this is going to be ATP, and we're going to move a solute against its concentration gradient to where we need it to be. So now we are moving upstream, I guess you could say, against the concentration gradient. Our job is harder. So there's already sodium out here, lots of it, but we want to get this sodium out of here, so it's going to take energy. So we load the sodium up into the channel. Sodium potassium pump is what this channel is going to be. We use our energy from ATP to push that sodium to where it doesn't want to be. We get it out of there. We have now our inorganic phosphate bonded to this because we are ADP now. And some potassium comes in, and we bring potassium in. So 3 sodiums out, 2 potassiums in. That's the basis for a sodium-potassium pump. And we can keep doing this, but it's going to cost us. Every turn of the cycle is going to cost us more ATP. So it's worth it because you need this for, like, I don't know, nerves to fire, which is awesomely important. You want to have nerves to fire, but it's going to cost you to make that happen. So membrane trafficking. By processes of endocytosis and exocytosis, cells can take in and expel particles that are too big for transport proteins, as well as substances in bulk. So what this means is sometimes you get something that's bigger than water and bigger than sodium and bigger than glucose, but you still want it in the cell. So if it's too big for a transport protein, you just eat it, the whole cell. You just move the whole plasma membrane in and make a big old vesicle and just take a big bite of the world around you. And that's how you move these things into the cell. So we are going to form and move vesicles. And the cool thing about vesicles is they're made of the same exact stuff that the plasma membrane is made of. And so we can make vesicles and fuse vesicles, and our cell's not even gonna miss a beat. It's great. So these are gonna involve some motor proteins, because once we get this giant bite of the outside world into the cell, we're gonna have to move it around, and it's gonna require ATP because we're doing some serious moving in the cell. So there's 2 basic types. There's exo and endo. Exocytosis means to exit the cell. Cyto, cell. Exos, exit. To leave the cell. So we're going to take something we don't want in our cell. We're going to walk it over to the perimeter with the vesicle, and we're going to fuse that vesicle with our cell membrane, and we're going to kind of spill the contents out into the extracellular matrix. And that's it. So endocytosis is the exact opposite within the cell. So we form a vesicle from the cell membrane, and we enclose these materials, like maybe some food or something good that we need in the cell, and we bring that into the cell. Those are the 2 big types of transporting something within a membrane that's large. So here it is. Exocytosis, we got some bad stuff we want out of the cell. We move him over here. He fuses with the membrane, and this stuff gets out of there. And then this vesicle just becomes part of the membrane, which is super cool. Here, pinocytosis, this is kind of like we're just going to take in a little drop of fluid. It's like taking a little drink. If there's stuff in that fluid, that's fine, no big deal. And so the cell just kind of makes itself a little Pac-Man mouth. And we get a little water in there, we get little solutes in there, pinches that little membrane off right there. And then we've got a vesicle. We'll take him in and we'll get some fluid, digest that stuff, and we're in good shape. And then there's receptor-mediated endocytosis. This bottom one's a little trickier, but cool. You get these little surface receptors, all these proteins that we've embedded in our plasma membrane through these long, arduous processes of transcription and translation and transport through the endoplasmic reticulum and Golgi bodies and et cetera, and we finally got them in where they're supposed to be, and they get to do their job. Hooray. So these little receptors sit up there, and something bonds them. There's lots of solutes here all over the place, but only red solutes fit these receptors. And when the receptors are full, we pinch in a vesicle, and we only have red receptors. Very cool. Could be some charged ion that we want, could be a big chunk of food, could be a bunch of viruses, and now we're going to get sick, could be anything. But that's a little more picky version of pinocytosis, where we just grab whoever's around and pull them in the cell. So very cool ways to transport stuff. So I just kind of explained these to you, but we'll go over them anyways. Pinocytosis, bring a drop of extracellular fluid along with whatever particles are floating in it, bring it into the cell. Receptor-mediated endocytosis, specific molecules bind to the surface receptors, then we bring them in the cell. And then finally, phagocytosis, eating another cell is what that means. So we get a large target particle, like a microbe or cellular debris, and we extend out some big old pseudopods, and we merge them together as a vesicle, and we eat this big old thing. Can you think of a cell in your body that would want to eat other cells? Hopefully, you said white blood cells, right? Or leukocytes or something like that, because at some point in your childhood, I hope you were watching Magic School Bus and they shrank the bus down real tiny and went into some kid's body, and then white blood cells attacked them and the kids were all freaked out. And I know that is stuck in your brain somewhere. So now you're just old enough and smart enough to call it phagocytosis and relate that to what you already knew about the Magic School Bus. So, recycling membranes. Exocytosis and endocytosis continually replace and withdraw patches of the plasma membrane, but like I said, it's really not a big deal to fuse that back in with the membrane. It doesn't affect the cell too much. And we're always regenerating new lipids from the ER and sending those out to make brand new cell membrane that fuses in. So that's important too. We're regenerating that bit that we're taking away through the process of endocytosis. So here's how you form a new plasma membrane. You get products made in the rough ER with a few proteins. These vesicles move into the Golgi. The Golgi processes them, releases these vesicles out. The little beginnings of proteins are now the fully functional quaternary form of the protein. This lipid starts to grow around the outside of this membrane, and then it fuses with the existing plasma membrane, and we have just replaced a section of fencing basically on this plasma membrane. And it lets out a little bit of fluid, but it also has rebuilt this big section of plasma membrane complete with receptor proteins on it, which is nice. So a lot of information coming to regulate all that, but we could certainly rebuild those. So this is the end of the chapter, and they end it with a few points to ponder. So back to the very beginning of the chapter. What factors do you think could be involved in how much alcohol each individual can process? There's some urban legends, I think, and old wives' tales about that, so it'd be kind of interesting to see which of those are true and which aren't. Why is heat the most common form of entropy? And what do you think would happen if our body pH rose to 9.5? So take a minute, scribble your thoughts down on those 3 questions, and we'll visit about them in class and see what we can come to figure out. So thanks for your time, guys. Have a good one.